Okay, so stable isotope probing. You guys have heard quite a lot, I presume, in various classes about radioisotope probing, right? And I just mentioned it myself. I mean, you put in a radioactive compound and you see where it goes. Radioisotope labeling and tracing is used a lot because it's really easy to measure, right? You've got a scintillation counter or even a Geiger counter, and it just counts radioactive decays. And you'd be amazed at how accurate you can be with radioisotope counting. We used to make tRNA substrate for RNase-P assays, and we could measure the amount of substrate we made, radioactive pre-tRNA, to within 1 part in a million. The concentration of an organic molecule, to 1 part in a million. Not so easy to measure some other things, so our measurements weren't that good, but that part of it was really easy. The problem with radioisotope probing is that in a particular molecule, for example, a ribosomal RNA, you're probably, if you make it radioactively labeled, probably only 1 or 2 of the phosphates, usually it's P32 people use, only 1 or 2 of the phosphates are actually labeled. If you labeled all of them, every time one of those atoms decayed, it would break the molecule, right? Imagine that in a cell. That would, the cells would instantly die because, at any particular moment, a bunch of phosphates are going off, and so it would kill the cell. And so when you radioactively probe, both the advantage and the disadvantage is that a tiny, tiny fraction of the atoms are actually the radioactive isotope. Stable isotope probing is the opposite. In the stable isotope probing experiment, you can load in nearly all of the stable isotope labeled material you want. And so, for example, you can buy glucose where all 6 carbons are C13, the heavy stable isotope of carbon. Not so cheap, but you can get it. It's not so bad. And that means that if you feed E. coli the C13 glucose, you can label all the carbon in the cell and have it heavy isotope labeled. This is how the Hershey and Chase experiment worked years ago, where they found out how semi-conservative replication worked in DNA, right? You guys learned that experiment long, long ago, I hope. Well, we're going to use stable isotope probing here. And so the trick is that if you want to find out what an organism can do, you feed it the substrate for the reaction you're measuring and allow the cell to be labeled by that. Now, you can't do every experiment this way, but generally speaking. And so, for example, if you've got a wastewater treatment facility where phenol degradation is happening, this is all the time. A lot of industrial wastewater treatment facilities have to deal with phenol. And you want to know who's eating the phenol, you would take a sample of that and you would feed it heavy isotope labeled phenol. So this is phenol. It's got 6 carbons. You make a heavy isotope labeled phenol where all 6 carbons are C13. You feed it to the sample, and the organism that can degrade phenol takes it in, and a couple of things happen. Most of that phenol gets burned off, it gets oxidized as CO2, right? And that floats out of the sample and you never worry about it. Does CO2 have any information about who did it? No. There's no phylogenetic information in CO2. But the cells grow too, right? And they build up biomass, and that biomass comes from the food they're eating. And so before too long, the proteins, the fats, the nucleic acids in that organism start to accumulate C13. Those molecules do have phylogenetic information. And so, for example, you can take the ribosomal RNA from those organisms and you see that they're labeled in C13. You can separate C13 labeled ribosomal RNA from regular ribosomal RNA. There are enough carbons, of course, in that RNA that are going to be labeled that the mass of the RNA will be increased. Every C13 in there is another Dalton to the molecular weight of that RNA. Now you can't do this with radioisotopes because you might have one, extra couple of Daltons in a ribosomal RNA, and that's not enough. But you could load a ton of heavy isotope into a ribosomal RNA, enough to physically separate it from the others. And if you can separate it, then you can pull it out and sequence it, right, and see who it is. So this is how the actual process works. You grow the organism with your heavy isotope labeled substrate. How long you grow it is a thing you have to test experimentally, because if you don't do it long enough, then not enough of the label is in the molecules you want. If you grow it too long, if it's a mixed population, that C13 starts to make its way through the food chain and other stuff gets labeled. Once you've labeled up your sample, then you separate them based on density. And this is done in cesium tetrafluoroacetate gradients. Have you guys learned about density gradient centrifugation at all? Yeah, good. And so this is a cesium acetate gradient, and it's light at the top and heavy at the bottom once you run it. The centrifuges that run these things are amazing. Imagine a rotor this big around made out of titanium or aluminum spinning at 100,000 RPM. Now imagine that failing. That's pretty awful. I had that happen once, by the way, as a grad student. Got a call. Jim, your centrifuge just exploded. It had quite literally exploded. There was a crack in the bottom of the rotor where the tube sits. Hairline fracture, completely invisible. It had been inspected the day before, and the technician hadn't seen it. The entire sample in that bucket squeezed through that crack, including the polycarbonate plastic tube it was in. Unbalanced the rotor, rocked off the spindle, came off, and blew the doors off the machine. It was traveling relatively slowly, 28,000 RPM, which is still supersonic. And so it made quite a noise. I've got bits of aluminum left over from it. This is military-grade aluminum. It was twisted up like taffy. It's amazing. Be careful when you run these things. The reason that happens, by the way, most often is because when you start this gradient out, this is a homogeneous solution. And in the case of RNA isolations, the density of RNA in the presence of cesium atoms is about 1.7. People often think that DNA has a density of 1.5, and RNA has a density of 1.7, and protein has a density of 1.1. That's not true. All those things have a density of 1, more or less. But they bind different amounts of cesium, and that cesium increases their density. RNA binds a lot of cesium. And so you average this out to 1.7, and when you spin it, the cesium atoms settle a bit down. And so this might only be 1.6 up here, it's 1.7 here, and it's 1.8 down here. And it's a smooth gradient. The danger is if the solubility of cesium is exceeded at the bottom of that tube. Because the tube starts out in solution, you're pretty close to the solubility limit. If you exceed the solubility where you've concentrated it here at the bottom, that cesium will come out of solution and forms a weight on one side of the rotor. It throws the thing off balance and causes a disaster. But after you've created this gradient in the centrifuge, the molecules will separate out according to their density. That is, an atom or a molecule of in this case, this is DNA, but the same is true for RNA. If it starts out in the tube down here, and the density starts to go up, it's going to float, right? And a molecule that starts out up here, as it starts to get less dense, will sink. And it will form then a band at its same density, where it neither floats nor sinks. And because you add ethidium bromide to these, which binds DNA and you can see it, you can go in, quite literally with a syringe needle and suck that band out and do stuff with it. Or you can poke a hole in the bottom and very gently let this material drip out and collect fractions. Once you've done that, you can do, if this is ribosomal RNA, you can do reverse transcriptase PCR and then run a denaturing gradient gel. And so this is this is a gel of the fractions from one of these tubes. And so this is the top fraction with the bands, with this band in it. These are two intermediate fractions, and this is the bottom fraction with the heavy stuff in it. And you can see these populations are different, right? And so this band 2, for example, isn't very much of it in the light fractions, but it's highly enriched in the dense fractions. Therefore, this organism is probably eating phenol in our hypothetical experiment. Likewise, band number 1. Band number 3 may be as well, although it doesn't show up in the densest fractions. Whereas organism number 4 here is prevalent in the light fractions, and then it fades away. This one up here too, etc. And so you can measure which ones they are, and then cut those bands out and sequence them and see what they are, right? And then do the phylogenetic analysis. So let's do an example of this.